about a page and a half. But it takes a lot of steps. I'll start them over here. We're going to go step by step what happens. Step number 1. The body cell presents the foreign antigen, meaning the foreign shape. By the way, a shortcut to say the body cell is an APC, antigen presenting cell. But you can just say body cell. I'm cool with that. So a body cell is presenting this foreign antigen. Where's it presenting it? On the what? On the MHC, on your self-antigen. So the bad guy thing is stuck on the my thing. How's that for scientific sounding? So I put the antigen on my MHC1 type protein receptor. Who is this? Who's looking at it? T cell. What kind of T cell? Nope. Nope, that's a chemical. That is a special kind of T cell called a helper T cell. Step 2. A helper T cell. That's one version of a T cell, also called the TH cell. Quote, checks the presented antigen. So this helper T cell is now going to look at, check, verify, whatever word you like, that red spot to see what it is. Because I have to learn what it is without killing myself. All right. Then the cells, the APC and the helper T, exchange chemicals. So we're going to do some signaling back and forth. Can anyone guess the name of the chemicals they exchange? They're both white blood cells.
Interleukin.
Interleukin, between the whites. So they're going to speak with interleukins. So if you ever read an article about interleukin blah, 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 they simply mean the chemicals that the two cells are using to talk to each other. Right? I have to talk to you, you have to talk to me. So the APC announces the trophy. Here I am, look at this. The helper T cell checks it. They do some communication, memo, whatever. And then, what's going to happen after that? Glad you asked. Let's follow the arrow down. See that big word in the center there is called proliferation. So we're going to have proliferation. Step number 4. The helper T cell orders proliferation. Proliferation of green here. Ah, excited. Orders proliferation of cytotoxic T cells. Okay. One thing that's weird about T cells is different types. So here the helper T cell is ordering proliferation of a different type called a cytotoxic T cell. What does cytotoxic mean in English? Cell killer, right? Cytotoxic. Okay, but let's walk backwards. What does it mean to proliferate? If I order you to proliferate, what are you going to do?
Get bigger, grow, make more.
You're going to grow, make more, clone. So you can substitute the word cloning. You can substitute mitosis from Biology 112. You're basically going to tell the cytotoxic T cells to reproduce. So in the military, who gives orders?
Officers.
The officers, the general. So the helper T cell is the general, the admiral, whatever you want to call it, giving orders to the cytotoxic T cell. So if we do our little kind of analogy, we'll call these, I don't know, the green beret type cells. Right? Assassin cells? Are there assassins in there? So the general is going to order the assassins to duplicate. Why do you want more assassins? If I can kill the thing, what am I going to kill?
The enemy.
That thing up there. So if you look at the shape of these cytotoxic T cells, I'm going to show you right now. You notice the cytotoxic T cell, here's one. Notice what it's fitting. It fits that shape, the original shape it found. So I had to teach it what shape to attack. So I'm only going to attack the shape I saw up here. That's what makes it specific. So the only shape you're going to attack, Mr. Assassin, is this antigen right here, the one you were told about in step 1. So if you had a different antigen, you'd have to re-announce and reorder every time. That's why it's specific. The cytotoxic T cells would proliferate, and they'd only be able to kill the shape in number 1. But we're not done yet. We haven't done anything. All we've done is made a bunch of military guys. Step number, what is that, 5? Mm-hmm. Step 5. Step number 5. The cytotoxic T cells will then locate and bind to anything with the foreign antigen. So anything that has that shape in number 1, I will stick to, I will find, I will locate you. I will seek you out. And then what does a cytotoxic T cell do with these things that have the foreign antigens? It kills them. So any cell with the foreign antigen, any cell, will be killed. Because that's what I was told to do in step 1 and 2 and 3. So if we look at that statically, here are my cytotoxic T cells. There they all are. Notice they have a shape. Hey, here's somebody with the antigen inside them. What am I going to do to this nice little cell? Destroy it. But that's my cell. Yes, it is. But we had to destroy the village to save it, right? That's the analogy. So even though that was a body cell, it's not autoimmune. You're not just attacking it because it's you. You're attacking the thing inside of it. So you just destroyed your lung tissue to get to the tuberculosis that was inside of it. So you're destroying your own body to kill the thing in the cell that had the antigen. Collateral damage. I blew up the building that could see I was a machine gun. Could have a viewer there.
So even though TB is in your lung, you still have that I'm going to kill you in your body. So
Yes.
if you get a TB virus antigen, you put it in your arm, it's going to get inflamed, and then hopefully it's going to go away. But if it doesn't,
that means you're already exposed and you've already been taught. That's how that test works. If you've already been exposed and taught this, then it would react differently than if this is what they call a virgin exposure. Okay. But the reason you get TB is you destroy your lungs, they scar over, to protect you from what you just did, and then you keep doing that over and over and over. And the bacteria then scars over to hide. Right? Yes?
With the TB test,
Yeah.
you said that it's usually no reaction because it's a virgin exposure. But like, when you're in medical field, they test you constantly.
Yes.
So isn't that starting to cause your body issues?
No.
Should.
Add it in
Such
there,
a
add it
small
in there,
amount.
add it in there.
Shouldn't.
It shouldn't.
Now, if you were exposed in your line of work and then you got a big exposure, that would cause it. But under those circumstances, it shouldn't. So the trick is, next time you have the flu and you feel so great and achy and you're coughing up whatever, just look at this picture and think about what's happening. The flu got into your respiratory system. Your body's doing its job. It's trying to kill the flu. The only way it can kill the flu is to kill the tissues it's living in, like your lung lining, like your joints, like whatever. If it works, if you survive, you would have killed off all the flu because you killed the cells that would reproduce the flu. Remember, biology 112 viruses require a host. If I kill the host, I kill the parasite. The problem is you feel like you got hit by a bus as your immune system's basically destroying you. The flu didn't destroy you. Your immune system destroyed you getting the flu out. But it works very well if you live through it. It's that if part there. But wait, we're not done. Because there's a step 7. Some of the cytotoxic T cells, some of them, not all of them, some of them, don't attack. They don't kill. But stay inactive. Why do you want some of them not to go out to the front and shoot people?
So the next have to reteach them.
I don't have to reteach them, but why would I need them again?
Because you have to.
These infections with that.
Very good. So we call these the reserves. You think about it, you send all your troops to the front and they all die, you have nobody. So you're going to keep some troops behind, call them the reserves. The fancy word you're going to say are memory cells. What do memory cells do? They remember. Remember the learning thing? They remember what you attacked the first time. So once I kill off the flu, some of my T cells will remember it and kill it the next time. So I don't have to constantly be relearning the same thing. I learned it once and remember. Make sense? Yes?
So that cell just hang out for, I mean, it couldn't really hang out that long, could it? Like
We
5
don't
years?
know how long they live, but they think it's decades.
So every time you get the flu, you're getting a new,
Yes,
completely new.
yes. Because if you had the same one, you wouldn't have gotten sick. So, your mom was right. You'll never get the same thing twice. The problem is, if it's not the same thing, you will get it. But you can only remember the first one you saw. So since flu changes, you have to re-remember it.
What about the flu?
And well, the thing, it keeps changing every 6 months, right?
So then, how do they choose which strain of the flu they're going to vaccinate you with?
Oh, that's a fascinating story. You go to Hong Kong, you find the 3 flus that are most common and everyone dying in Hong Kong, and you say that's the one that's going to be in the U.S. 6 months from now. You fly back to Atlanta, CDC, you make a virus vaccine really fast of the 3 that you think will be here, and you hope like anything like going to Vegas, it's the right 3.
Because they all start in Hong Kong.
They all start in Asia. So you go to Asia, you predict 6 months they'll be here, you try to guess what it's going to be like 6 months from now.
No, why do they all start in Asia?
Because people live with their pigs.
That's gross.
So if we didn't live with pigs, we'd have less flu.
Yes.
We live
And birds.
with pigs forever.
So we should go to Asia and kill all the pigs.
That's a great idea.
I'm sure China would love that concept. But no, one of the problems with Asian culture countries is there's a lot of people, 1 billion people, right? Lots of pigs, lots of birds.
And they all live with
And they all live very much, not necessarily with each other, but crammed together. So a pig can be infected with birds and human viruses. What they call transgenic shift, meaning that now you have a people virus that's now like a bird virus. Your body's never seen a bird virus. So if you get exposed to the pig, it now has a human-bird hybrid that you've
That's
never
like how we
seen. So
get
you
all
got to
these
then run through the
Weird
batch here.
things
Man,
from
they're
monkeys
big.
too.
Yeah, but usually there are not as many people around monkeys. That's the thing. China has 1 billion people all within spitting distance of each other.
Unless monkeys throw their poo at you.
Yes, so hopefully you don't. Yeah, what are you-
So why don't we have the problem with those type of infections in, like, say some of the Appalachian cultures where they have the chickens running in and out.
Usually not that many people, and they're spread away from each other. One of the problems with Hong Kong is the bird flu thing in Hong Kong, right? You have, 25 million people all crammed within a 10-mile square spot. So all it takes is one person sneezing, and immediately everyone gets it. If you're in Appalachia, yeah, you may die, your family may die, but it's 3 miles to get to the next person.
So it's more of the population density.
Combination density and also cultural habits, how much exposure you have, hygiene, all that stuff. Yeah.
There's the
Go.
okay.
Just
There's
waiting for a while.
viruses that come back up.
Yes.
Like you
Ebola.
get shingles. I'd probably get shingles if I were to have a relapse or whatever you
Yes.
call it for. How can my body make that not happen?
You have to keep your immune system up to speed. What happens normally in shingles is you got chicken pox when you're little or something. It went away, your body remembers it. One day you get old, you get run down, you're not eating well, you catch 5 other things, your immune system's busy with those, forgets all about the shingles. It's a 2-front war, right? I can't fight the flu and the shingles at the same time. So you get immunocompromised in some way. The shingles, which you remember, can come back before you basically can fight it off. So older people tend to get more shingles because A, they're immunocompromised, poor health, it's been longer since their exposure, all that adds up to more likely to get it. As long as you're in good, as long as you're not taking this class and not stressed, you'll remember it. As a teacher, I always catch the flu in finals week. As I get more stressed, my immune system is, remember
Why are
sympathetic
you
nervous
stressed?
system, right? Shuts off immunity. These guys all go on R&R, forgetting their job. Then the flu gets ahead of them, right?
So when does the symptoms start to show up? In which number?
Depends. First exposure, you've already got the symptoms long before you got like to number 2.
Oh,
Second exposure, you won't have symptoms because you'll be at number 7.
so what's the difference between first and second exposure?
Is if you have the memory cells. Once you have memory cells, you've already been exposed once. You can basically skip 1, 2, and 3 and jump right to number 5. It's a quicker response.
So how come people, like, I've never had the flu before, like, how
You're
come?
kidding me, right?
I've never had the flu. And I never get flu shots, so I've never had it. And how can people, I mean, like, how does that
You should
happen?
donate your body to science right now.
Oh my god.
I've never had it.
So you've never barfed ever?
You had a fever?
Well, I've had a fever, not for a really long time, but no, I've never thrown up from being, like, sick.
Or like sneezed or had a runny nose?
Not really.
Holy cow.
Good for you, though.
I just don't understand how that happened.
I don't understand how that happened either. You just have a really great immune system, or you don't get the flu, or you get,
Is that bad?
No.
Like, I mean, I know other people that don't really get sick very often.
They have
so
really
that's
good immune
why-
systems,
yeah,
or
okay.
you've been lucky.
Are you just, like, born like that?
Do these cells transfer from?
Nope.
I mean, is this like a?
Unless you
From
get
mother to-
immunoglobulin. So the Ebola case, for example.
I mean from like mother to
Get that
fetus.
cell. But if you
Do
transfer
you have
blood
kids?
from somebody,
That's probably
then
why.
you can transfer the white blood cells with them.
I got
The
really
Ebola
sick
thing, you
when I
find
was
someone
kids.
who's alive, you take their
Yes.
blood out of them,
Especially when they're
you can
in
transfer the white blood cells.
preschool
But a mom
or
baby thing,
kindergarten.
that's not good.
What about ones that are dead?
Though they're dead, you can't do much with that.
Yes, huh, because they're actually more contagious when they're dead.
Because they are, so you don't want to take their blood, you want to take someone's
Oh, I
alive.
thought you, okay.
No, not yet.
First part of the
No.
conversation, my bad, sorry.
So theoretically, we could do like a complete dialysis experience with somebody with some bloodborne illness and give them good blood and hope their body takes.
Yeah, you could. Those are the details.
Oh.
So let me show you the steps so far. So here is my body cell pick a word. I don't know which word you want.
Activation of the immune response typically begins when a pathogen enters the body.
Flu, Ebola.
Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces.
That's step 1. I have to show the immune system someone's invading. You got to sound the alarm. Right? You got to do sentry duty, whatever your analogy is. But that's APCing. I have to present. So here's the bad guy on my uniform. Right? That has to happen.
Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells.
How about that?
An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen.
Here's the helper T cell, the general, the boss, pick a word you like. He, she, it has to check that and verify that that's not you and something you want to do. So we're having to train.
And
Yes.
ATC is always a macrophage?
Normally it's a macrophage. It could be any cell. Macrophages are the number 1 because they're the ones that are going to be eating stuff and presenting it 90% of the time. But any body cell could present. Just they're usually first one.
During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2.
Between the whites, right?
Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells.
We'll do the B cells a little bit later, but here are the ones we're looking at. The cytotoxic Ts. We're turning on the army. We're turning on the ninjas, the assassins. Pick a word you like. Right? Because we want them to go kill. The helper T cell doesn't kill anything. Officers never go to the front, right? We just give orders. So we're going to tell the troops what to do. So here we go.
The immune response from this point follows 2 paths, one using cytotoxic T cells and one using B cells.
By the way, you'll normally do both. You will eventually do both B's and T's, but this animation will take you through T's first. But you could be doing the B cell the same time you're doing the T cell. Perfectly allowed. In fact, you want that.
Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen.
Specific.
The
Right?
cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen.
So that's basically step 5 and 6. So I find whoever has that red dot that my boss told me to kill, and I just kill you. One or the other. But now you have died, the thing that's red inside of you has also died. But some of them will stay behind and be ready for the next round. If I get infected a second time, they will then be able to attack without having to go through all this long-winded process. Makes sense?
Oh my gosh, oh yeah, are we gonna get into the chemistry of like what chemicals they release to kill that cell?
Yes, we can. Let's put that on the board. That's actually a chemical PC says thou shalt know. Kill cells! The magic word, you'll love this name. It's called perforin, or perforin. That's a protein. What do you think perforin protein does? It perforates. It basically blows apart your lipid bilayer so you leak out. So you punch a hole in the cell, and the cell just basically leaks its guts all over you. So you're going to basically secrete perforin. That's what the stuff was there. The cell deflates, taking the infection with it. But some of the cells stay behind. That's the T cell response. I'm going to kill things with a foreign antigen. But we're also now going to do the B cell half. All right? So the trick with the B cell is it's the same general idea. It's just a different cell. So here we have some B cells. Looking at them, the big yellow guys, what do you notice about their surface?
They
Let's
have
see.
proteins.
They have little things, right? What do you think those things are? Those are receptors or proteins, pick a word. All right, so what do you think those receptors are? Ooh, hey, here's something. Here's a bad guy with its antigens. They say that's an antigen receptor. Can you guess what we're going to do now? Receive and join. Okay, which one of these 4 cells will best grab the bad guy? This one that's labeled. How about that? Why? They match. So one of the concepts here is this is all shape dependent. I look at the shape of my antigen to learn what to kill. So same thing with B cells. They're shape dependent. I can only match a shape that you tell me to match. That's why it's specific immunity. Right? Let's go through the steps to get these guys to go. So in the B cell system, you still need the same general steps. You have to have an APC. The helper T cell still has to check. And step number 3. So these first 3 steps are the same in T and B. The first 3 steps do not change. You have to activate your helper T cell before either system comes on. So you're just going to repeat those 3 steps a second time. Where it gets different is step 4. In step 4, the helper T cell would order proliferation of what? They wouldn't be cytotoxic T cells anymore. What are they?
B cells?
B cells. So where things begin to go differently are step 4. We're now going to turn on the B cells. In the prior one we did T cells. These are going to be B cells. So we're going to turn on B cells. And that actually changes over here. So I'm going to proliferate, clone, reproduce, mitose my B cells. Is this step 5?
Yeah.
Yeah. Okay. Some B cells become what they call plasma B cells. Where do you think plasma B cells would be? In the plasma. Let's look at them. Here are some of my plasma B cells. They look just like the other B cells. What do you notice about their shapes, though?
The same.
They're all the same. Remember, cloning in Biology 112 was making the same. I'm going to make the same B cell as the one over here that could match up, so they're specific. So these are my B cells that I'm cloning. Then I want you to look what they're going to do. Here's a picture. So here are my B cells at the top. They got told by the helper T cell to clone or proliferate, reproduce. And some of those B cells become plasma cells, right here. And they're making something. What are they making?
Antibodies.
Antibodies. So plasma B cells make antibodies. Not antigens, antibodies. Remember blood typing? You learned antibodies, antigens.
That's step 6 or that was?
That's still step 5. So the plasma B cells make antibodies. So what we need to do now is figure out what you do, why you want antibodies. Let's find our antibodies, which are right here. Antibodies stick to antigens. Or bind to. And this is what you learn in blood typing. Remember, your blood had antigens. You had the antibodies to make it clump. Same thing here. So you're going to make antibodies to stick to, and whose antigen are you going to stick to? The very number 1, the one that you saw the first time. What happens when you stick to an antigen? I'm glad you asked. Look at this picture. This is saying my antigen gets stuck to my antibody. And I could do a variety of things. I could surround you so you can't hurt me because you're away. Here's what you did in lab. I can make you clumpy. What do clumped things do? They don't go anywhere, right? So you're stuck in the mud. You can't go, in fact, you can't get anywhere. Or I can make you basically go in my pee. No matter how I do that, or I can poke holes in you. If I plug you up, make you clumpy, punch holes in you and hold you down, at some point, somebody's going to eat you, or you're going to die. But you're not going to hurt me anymore, because basically I trapped you in the barbed wire at the front, so you can't advance. So the goal of the antibodies is to prevent the pathogen from moving. The barbed wire of the First World War was this idea. If you get trapped in the wire, I can kill you any old time I want. And that's what you're going to do with the antibodies, is you're going to stick to the antigens and hold them down to be eaten, beat up, killed, stepped on, whatever. Yep, spider web. Same concept. So the antibodies technically don't kill anything. They just hold you down. Flypaper concept. The flypaper didn't, I didn't kill the flypaper killed the fly. Those little mouse traps, you stick their nose down and get stuck, and you just sit there and laugh at them. That's this concept. Right? I didn't kill the mouse, he just suffocated for 2 weeks on the little flypaper thing. That's what you're doing here, is your B cells are making antibodies that trap the antigens. Right? And then the white blood cells can clean up later. Make sense? Okay, so PCC says that's not enough to learn, because you're supposed to know where different antibodies are found. Take a picture of this with your phone. Okay. Thou shalt memorize the different classes, functions, and locations of the antibodies.
Where can you find this in the book?
Somewhere. I honestly don't remember the page. But to make this make some sense, you can also find this on Google if you type in antibody classes.
You say structure as well, or can we skip
No,
the
structure, I don't care how you draw it. Please don't draw it. By the way, antibodies are abbreviated IG. Immunoglobulin. You can say antibody, but on this chart it's IG something. And they all have letters. Here's IgD, IgM, IgG, IgA. These are different types of antibodies, all made by a B cell. We're going to put them over here on the big chart.